All right, we are moving forward with transcription. We've completed DNA structure, we've completed DNA replication, what that process looks like, where it takes place, when it takes place in the cell cycle. And now that we have our DNA replicated, we have to transcribe it. So the process of transcription after replication is converting a gene into a single-stranded RNA molecule. This video is important to know and understand and to watch. You have to watch this. It might not make sense right now, but as we go through transcription and we do a little practice with it, as we go through translation and do some practice with it, it'll truly make more sense. And it's a good learning process for you. It's good to see it happening and hear the vocab words that you now understand. So take the time to watch it. Okay, so transcription now is kind of the second stage, or it's the second process after DNA replication has occurred. The central dogma is stating that information is flowing from DNA to RNA to a protein, and that's what this whole chapter is about, DNA to proteins. And so we've made that extra DNA, that second set of DNA, and now we have to make RNA. That's the red that you see. This is still taking place, also, you can see, in the nucleus. So here's kind of our process. Blue is checked off. We understand DNA, we get replication and that whole process. And now, since the DNA is replicated, we are going to talk about the process of transcription. It's kind of the link between DNA and making a protein. So when we talk about transcription, we're going to be talking about RNA, which is a ribose sugar instead of a deoxyribose sugar. That's what DNA stands for, deoxyribose sugar, where RNA is standing for or has a ribose sugar in it. RNA has uracil. DNA has thymine. And so when we talk about this process of transcription, whenever there is an A that is exposed, in the process of transcription now, the complementary base is going to be U. There is no more thymine or T in the process of transcription. T goes away. And I'll give an example when we talk about that process. And then finally, RNA is going to be single-stranded. DNA, there was a double strand. RNA now is going to just be single-stranded. So this is a good thing to write down and compare and contrast DNA versus RNA. Could be a short answer question on the test, never know. All right, the three types of RNA that transcription makes are messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA, or mRNA, is going to carry the message that will be later translated to form a specific protein. So once there is an RNA strand that has been transcribed, mRNA is going to pick that RNA strand up and take it away to be translated to form a protein. So it's carrying the message. Transfer RNA, or tRNA, is going to, tRNA is bringing amino acids from the cytoplasm to the ribosome. This actually takes place when we talk about translation, but we have to understand that transcription is making these types of RNA. And then finally, ribosomal RNA is forming parts of the ribosome where the actual protein is going to get made. So make sure that you don't just know that there are three, make sure that you understand what each of the three do. Okay, so the process now is as followed. So what we're making at the end product of transcription is going to be a single strand of RNA. I told my classes the whole process of replication, and then what is the end product? The end product is 2 strands of DNA. Half is going to be original, half is the new. Same thing here, transcription. There's a process, and what that end product is going to be is a single strand of RNA. And so what happens here is we can see the yellow shading in the picture. That's the transcription complex. It contains RNA polymerase, it contains proteins, and really what that does when that builds together, it's going to recognize on the DNA strand where to start this process of transcription and unwind a segment of DNA. That's kind of the beginning part of it. And then similar to DNA replication, nucleotides are going to pair with the strand of DNA, but this time it's only pairing with one side of the DNA strand. So any exposed bases, if there's any C's that are exposed, G is going to come in and bind together. If there are any A's exposed on the DNA strand, U's are now going to bind with A's. If any T's are exposed on the DNA strand, A's are now going to come and bind onto that DNA strand. And then finally, if there's any G's, C's are going to bind to that. So make sure you note the difference here. If there are any A's exposed on that DNA strand, U or uracil is now going to bind to the letter A. RNA polymerase is going to be what bonds the nucleotides together. So then the DNA helix is going to wind again as the gene then will get transcribed. Notice in the picture what's happening is that it's binding to one side and then it's also pulling apart right away so that the DNA strand can wind back up. And then finally, once the RNA strand has reached a certain point, it's going to detach from the DNA and it now has been transcribed. So you've got a single strand of RNA made up of A's, U's, C's, and G's. And what's going to happen is that messenger RNA is going to take that message and it's going to take it out of the nucleus to look for a ribosome where the process of translation is going to occur. Okay, so you have completed the 8.4 notes. If you need to go back, if you need to watch the video, if you need to pause, slow down, if you have to take a look on the online textbook, whatever you have to do to understand this information and this process, do that. Ask questions. Teachers will love to answer them for you. So take care of business. Next is to do the 8.4 walk-in quiz, and then you are done for the weekend. Take care.